Hi, my name is Matthew Harms, and I practice neuromuscular medicine and neurogenetics at Columbia University in New York City. And I want to thank you for taking the time today to listen to this talk about what causes FSHD. Now, when I interview genetic counselors, the very first question I ask them to see whether they're worth their salt is whether they can explain how FSHD occurs from a molecular perspective. I use this because it's a complicated test, because it's a complicated subject, but my goal for today is to break it down as simply as possible to help you better understand exactly what's going on underneath the surface of FSHD. So I didn't introduce myself with this clock running in the background for a random reason. It turns out that a watch mechanism to make sure that it's keeping time and doing a good job of letting you know how long I've been talking is a complicated mechanism. So if you peel it back from the surface of the watch, you get these complicated gears and springs and mechanisms all designed to work together in perfect harmony. If there's a little bit of dust between some of the gears, if the spring is too tight, if one of the pieces becomes loose, then the inside of the watch doesn't work and it doesn't keep time. In fact, it usually comes to a grinding halt. And it turns out that most of the cells in our body and our organs themselves are finely tuned clocks, just like this one. So the muscle fiber itself, the area where things go wrong in FSHD, is just as finely tuned a machine as that watch. Shown here in this very simplified diagram are some pieces of that complicated machinery. There's the contractile apparatus shown down there at the bottom, which are basically rods that have to slide past each other in perfect synchrony in order to cause muscle contraction. Muscle contraction changes the shape of the cell, so the cell has to be strong in order to withstand the stresses and the shear stress. So there are a bunch of other proteins that all line up and work together to make sure that the muscle fiber doesn't fall apart as it contracts. Each one of these individual parts has to be present, it has to be doing its job in order for the muscle fiber to be functional. And in fact, to stay healthy and run smoothly, a muscle fiber has to have all the necessary parts, the correct number of each part. Those parts have to all be in the right places, and there has to be a way to detect and replace any of the parts that might fail. Now, when we talk about a cell, and I use the term part to make the analogy back to that watch, what we really mean when we say parts are proteins. And proteins, of course, come from genes. How does this work? So shown here in this diagram is a chromosome, and across that chromosome are 5 different genes color-coded so you can keep track of them as I illustrate. So gene A makes protein A, and I've color-coded each of the proteins, which takes on a unique shape so that it can do its job. Gene B, C, D, and E all make their own unique proteins, and those proteins are the building blocks of the machinery that make each cell function. Now, at baseline, you don't want any genes to be turned on. You want a single cell to be able to select from the 19,000 different genes in our body to produce all of the unique protein machinery that it specifically needs. So, for example, you need myosin protein inside of a muscle fiber to contract, but a cell in the lining of your sinuses, for example, that's supposed to produce mucus doesn't need myosin, so it keeps that one off, but turns on the mucus-producing gene. So to kind of create a slate of genes to choose from, most of them are turned off in our cells. And then those genes do not produce their proteins. So how are genes kept off? Well, if you zoom in on gene B and gene D, if you want the gene to be silent, you attach specific chemical markers called methylation signals. There are other ways and other things that contribute, but simplistically, these methylation signals actually physically stick to the DNA and tell the cell not to use it. Methylation means the gene is going to be off. So there's methylation on gene B, and it's off. There's not methylation on gene D, and it's going to be available to be utilized. But it's not just enough to get rid of the methylation to turn the gene on. You actually have to actively turn it on. So here, all of the genes are inactivated and not being produced. And now the muscle fiber decides that it's time to do its job. And so to make itself functional, it knows that it needs to have a protein complex that looks like this, comprised of the purple yellow and dark gray proteins produced by gene B, C, and E. So the methylation signals get removed and an activating signal gets added for the genes that are necessary. And then those proteins can be manufactured and give rise to the protein complex that the functional muscle fiber needs. So how do these genes get turned on? Let's zoom in on gene B, and you can see here that at baseline, it's heavily methylated. Those signals are there and have it turned off. But if you want to turn that gene on, the first thing you do is you remove those methylation signals, and then you have to add the activation signal, the activating mechanism called a transcription factor. So in this case, the green transcription factor, a specific type of transcription factor, recognizes and sticks to a transcription factor binding site that is unique for that transcription factor. So there on gene B, the little green stripe is a region of the DNA that specifically attracts the green transcription factor. And now with the methylation removed, the transcription factor present, gene B can be manufactured. Protein B can be manufactured. No green transcription factor to bind, no production of protein B. Similarly, let's take a look at gene D. It has its methylation signals intact and a recognition signal for a red transcription factor. So to activate gene D, remove the methylation signals. And now if the cell is producing the red transcription factor, it will bind and stick, and now you'll produce protein D. No red transcription factor, no production of protein D. So the silencing mechanism of methylation and the activating mechanism of transcription factors work very closely together to make sure that the right genes are turned on at the right time. Let's take an illustration of what would happen if this mechanism was not working right. So to illustrate the point that inappropriate gene expression is bad for cells. So again, we have this muscle fiber that to be functional needs that complex of protein B, C, and E. So you need methylation at gene A and D, no methylation at gene B, C, and E, and the transcription factor for B, C, and E present to turn on those genes. Now, if that all gets messed up, so that abnormal transcription factors are present or abnormal methylation is present at gene E, for example, then you're going to have a different pattern of protein expression. Protein A and D will be made and protein E won't be present. And now the complexes are abnormal and can't be formed properly. So you'll end up with abnormal complexes and the lack of the healthy complex. And now you'll end up with a broken muscle fiber. So proper gene expression is absolutely essential to cells doing their job. And in the context of fascioscapulohumeral dystrophy, you need to have the right proteins, the right amounts of those proteins, and they have to be present in the right location and at the right time. To do that, you need the right transcription factors in the right amounts at the right times. And you need the methylation signals to be present or removed on the right genes at the right time in order to get the right protein production. And in FSHD, there's a disruption of both of these. Now, a lot of effort is being focused on the abnormal production of a transcription factor in FSHD called DUX4, and it's roughly akin to that green, those other transcription factors, the green and red stars. So that abnormal expression of the transcription factor DUX4 has downstream effects. It goes and it binds to the binding site, transcription factor binding sites in multiple genes across the genome, and produces and turns on proteins that aren't supposed to be there. Shown here are a few that people have been focusing on, and though abnormal production of those proteins leads to a sensitivity to oxidative stress or wear and tear, it leads to the inability of muscle stem cells to do their job and turn into new muscle fibers to replace damaged ones. It leads to abnormal inflammation, which can cause damage to muscle fibers, and activates pathways within each of the muscle fibers that eventually lead to and trigger cell death. And all of that comes together to give muscle weakness and the muscle atrophy that characterize fascioscapulohumeral dystrophy. So where does this abnormal transcription factor DUX4 come from? Why is it there? And what can we do about it? So DUX4 actually comes from the very tail end of chromosome 4. So this is called a karyotype. It's all of the chromosomes in a specific individual. And the DUX4 transcription factor is actually being manufactured from the circled region there, chromosome 4q35. So let's zoom in on what's there. So at the very end of chromosome 4 is a region called the D4Z4 repeat. It's comprised of repeating chunks of DNA that are pretty much identical one to another and are called KPN1 units. And buried within each of those KPN1 units is a broken copy of that DUX4 transcription factor. It's broken because it doesn't have an important signal it needs called a poly-A tail. That's a signal to the cell that it's a normal gene and should be made into a protein. And without it, basically the DUX4 is broken and it gets degraded. The RNA for it gets degraded and it does not lead to the production of DUX4 protein. But some of the chromosome 4s out there in the population actually have a poly-A tail. But under normal circumstances, it's too far from the D4Z4 broken DUX4 copy in order to activate the DUX4 protein. But if that poly-A tail gets close enough to the broken DUX4, now it gets converted into functional fixed DUX4 RNA, which does in fact go on to lead to the protein production of DUX4. And that toxic to cells like we talked about in the previous slide. To make sure that no DUX4 gets produced, the cell also uses those methylation signals to shut down and silence the D4Z4 region. So in normal individuals, this region has lots of methylation signals from stem to stern to make sure that even if that poly-A tail could get close enough, it would not result into production of DUX4. So what happens in FSHD? First, let's take a schematic look at the normal situation. So here is that D4Z4 region on chromosome 4q. It's heavily methylated and it's the proper size. So the broken DUX4 gene in the KPN1 units is too far from the poly-A tail, and so no DUX4 transcription factor is being manufactured. Now, in the most common cause of FSHD, there's a deletion of some of those KPN1 units. You can see that it's much shorter, and that poly-A tail has been brought closer to the D4Z4. And now, because of the proximity of the poly-A tail to the DUX4 broken gene, the DUX4 functional gene is RNA is manufactured, and the protein is expressed. That's what happens in FSHD type 1. Now, some people with normal size D4Z4 regions also develop FSHD. So what's happening there? In that circumstance, it turns out that there's an abnormally low amount of methylation across the entire D4Z4 region. And because there's no methylation, occasionally the cell gets a little mixed up, and it does manufacture full-length DUX4 RNA from that poly-A tail and produces functional DUX4 transcription factor. And that's what happens in FSHD type 2. FSHD type 2, 1 accounts for 95% of FSH, while FSHD type 2 explains about 5%. Looking over on the left side of the screen, just as a reminder, 4q35 deletions are the genetic cause of FSHD type 1, whereas mutations in other parts of the genome, in genes called SMUCHD1, LRIF1, and DNMT3A, are the cause of FSHD type 2. How do we genetically test for these molecular abnormalities? So we always begin by looking for the more common mechanism, and that's the 4q35 deletion. The standard test is called a Southern blot, and it looks for abnormal sizes of chunks of DNA. So shown currently on the left-hand side, there is a normal situation, and those little bars represent the normal bands of DNA and sized chunks of DNA. But in FSHD, if there's a deletion, then you end up with smaller chunks that you can visualize on that test. More recently, a technique on the right side called chromosomal combing has come up, where you can actually stretch out the chromosomes, and by looking at the pattern of genes and how far apart they are, determine whether or not this is an FSHD genetic mutation. And even more recently, something called single molecule optical mapping uses another technique essentially to stretch chromosomes and see what's present or absent, and can determine whether or not the FSHD deletion is present. That accounts for 95% and finds explanations for 95% of people who clinically seem to have FSHD. If the deletion is not present, or the contraction of the D4Z4 is not present, then the next genetic step is to measure the methylation status of the D4Z4. We know it's the normal length because the southern blot or chromosomal combing told us that, but does it have those repressive, silencing methylation signals? If it does, then this is not FSHD, and it's another muscular dystrophy that's mimicking or having a similar pattern to FSHD. If, however, you find that the methylation is reduced, then you go on to sequence first SMUC-D1 and more recently, with those discoveries of these other genes, LRIF1 and DNMT3A. Undoubtedly, there are other genes we don't know about yet, and so if any of those are negative, then getting to a center where those advanced genetic discovery research studies are going on would be advised. So that's how we do it currently, but a shift will be coming in the near future, and we'll be able to do a single combined test for FSHD using something called single molecule nanopore sequencing. This actually takes advantage of a really tiny little machine called MinION there that plugs into a laptop and can actually sequence extremely long chunks of DNA. So you can look at the entire region of chromosome 4q at a single go. And by plotting what's happening there, we'll be able to measure the size of the deletion if it's present, or see that there isn't one if it's not. We'll be able to detect the presence of that poly A tail to see if it's a type of chromosome 4q that can lead to FSHD. And we'll also be able to detect the methylation status and know whether FSHD type 2 or a combination of the two might be present. So let's put it all together. On the left-hand side is the most common form of FSHD due to a deletion of the D4Z4 on chromosome 4q35, bringing that poly A tail closer to the DUX4 gene. On the right-hand side, FSHD type 2, more rare, but due to mutations in genes that are responsible for controlling methylation at the D4Z4 region. Regardless of which mechanism is present in the cell, the production of DUX4 occurs when it's not supposed to occur. Now, across the genome, there are genes that have binding sites for the DUX4 transcription factor. So DUX4 goes to those genes and activates their production. The inappropriate gene expression that takes place then leads to a cascade of other things that occur, other genes that get activated, downstream pathways that get disrupted, and these go on to lead to the cellular damage, the muscle fiber damage that occurs in FSHD. So the good news, although damage is occurring and we aim to stop it, is that understanding this molecular mechanism has given us great ideas about what therapeutic targets might be present to design therapies around. So for example, if you could delete the poly A tail or delete the broken DUX4 copy using gene editing like CRISPR-Cas9 to go in and actually lop it out, then there would be no DUX4 manufactured. If you could stop the cell from using abnormally produced DUX4 RNA, then you wouldn't produce the DUX4 protein. And so in this circumstance, we would use something like antisense oligonucleotides to block the manufacture of the DUX4 protein. Or we could use antibodies that bind to the DUX4 protein and prevent them from sticking to their transcription factor binding sites. Also, if we could figure out which of the abnormal genes that are being expressed are the most important, we could design drugs to counteract those effects and block the downstream damage and the downstream pathway dysregulation that occurs from DUX4 expression. Of course, it could be that a combination of all of these are required, and for sure, additional research is needed. But the main goal is to intervene as early as possible so that we can restore the really complicated inner gears and springs of the watch that is the muscle fiber, so that it can go back to functioning, that it can stay functioning, and actually keep the accurate time that it needs in order to be a functional watch, or in this case, a functional muscle fiber. So with that, I will thank you again for your time and attention, and go ahead and open up the floor to answer questions that my presentation may have raised. Thank you.